Hey guys, welcome to the second set of our flipped classroom notes. I'm going to go ahead and get started because I don't want to waste too much time. Okay, so you can see this picture on your notes. What I do want you to do is I want you to write these words that are on the slide. If you want, you can go ahead and pause it now, write the words, and then listen to me as I speak. So I just want to get started by saying that scientists are very interested in how cell division occurs, how cells are able to start growing and stop growing. So what they do is they're able to actually grow cells outside of the body. So they can take any sort of body cells from a human body, from an animal body, and they're able to put it into a little petri dish here and feed it all its nutrients that it needs to divide. So what happens is once it gets all its nutrients, the cells are able to divide and divide and divide and keep growing and growing and growing until the entire petri dish has been filled. Once the entire petri dish has been filled, the cells will get a signal, hey, we don't need to divide anymore, we can stop. So then they go into interphase and they stop dividing at this point. Then what can happen is you can just completely remove the cells in the middle of the petri dish, and some of those cells will be injured, some of those cells will not be injured. All the cells that will be injured will go through apoptosis. All of those healthy cells around will get the signal, hey, we need to start dividing. So they start dividing and new cells get turned on. And then what happens is once those cells fill up the petri dish, they will stop dividing. They stop dividing because they come into contact with each other, and that is what we call contact inhibition. As soon as a cell touches another cell, it will stop dividing. Okay, and scientists have done that, and they have decided that there is a way to actually turn on and off the cell cycle. So they see that the cells in this petri dish don't keep dividing. Obviously, at some point, they stop. So the cell cycle is able to be turned on and off. Okay, so we're going to be talking about how that happens. Okay, so we have in our genes what we call cell division genes. And we know this because there are certain genes that can actually be turned on in case of an injury, in case of getting hurt, a cut, anything like that. And some cells get, after an injury, get stimulated to divide and heal and replace all those damaged cells. And once they completely replace those damaged or missing cells, then they are turned off when once the repair has been completed. And that cell injury is actually a signal. Okay, so once you get an injury, that is a signal to turn on those cell division genes. And then once the repair has been made and there's no more room to grow, that is a signal for them to shut off. Okay, and I know I've mentioned this before. Some cells divide quite frequently. Some cells go through the cell cycle very frequently. Some cells go through the cell cycle sometimes, and some cells actually only go through it one time. Okay, so an example would be human skin cells that divide very frequently, at least once an hour. I heard somewhere that you actually shed about 9 pounds of skin cells a year. That's pretty gross, but very interesting. So we know that your skin cells actually divide a lot, okay? And then some skin cells, or some cells only divide occasionally, such as liver cells. And like I said, you don't really do too much damage to your liver cells. So those only divide about once a year, just from the normal wear and tear. And then your nerve cells, you only have those nerve cells once in your whole life. You don't ever grow more nerve cells. You get the nerve cells that you get, and they don't continue to grow. So that's why a lot of spinal cord injuries are very, very bad. A lot of people get paralyzed, and there's no there's no cure really to that other than doing stem cell research, implanting different stem cells, trying to get them to be nerve cells, and they are definitely working on that, which is really great. Okay, so some scientists were looking at cells and wanted to figure out this whole regulation thing. So this started in the 1980s, where scientists actually discovered that there are proteins that cause dividing cells to form their mitotic spindle. Okay, and what happens is the scientists wanted to know what was causing the cell cycle to turn on and off. So they were able to so they discovered that there are certain proteins in a cell that cause the mitotic spindle to form. And they discovered this because they were able to actually remove part of the cytoplasm from a cell that was dividing, and they injected it into a cell that was not dividing, that was actually an interphase. And what happens is that cell all of a sudden started dividing right away as soon as that cytoplasm was injected into the non-dividing cell. So they decided that there must be some sort of molecule in the cytoplasm that is allowing for the cell to start dividing. And so we know that molecules, one of the most important molecules to life is proteins. And so what they did was scientists looked at the amounts of proteins that were present before and during and after cell division. So what they found was they actually found a certain protein that was very prominent during cell division and then not so prominent during interphase. And they called these cyclins because they seem to control the cell cycle. So that's why they call them cyclins. And there's not just one type of cyclin. There is a whole family of cyclins that seem to control and regulate the cell cycle. There's many checkpoints in the cell cycle that these cyclin proteins actually regulate. And they regulate the timing, like I said, in all eukaryotic cells. Okay, so talking about regulators, these cyclins are definitely regulators in the cell cycle, and they are in the cell, so they would be considered what we call an internal regulator. It's all regulators that are found inside of the cell, so the proteins will respond to any sort of signal that occurs inside of the cell, and allows for cell cycle to proceed only if certain processes have occurred. So there are checkpoints, like I mentioned, and those checkpoints check to see at certain spots of the cell division. Right? You can't go on to G2 phase until S phase has been completed, until the DNA has been copied. Then you can't move on to mitosis until all the nutrients are needed, all the organelles, until there's more organelles. So what happens is there are regulators that check to see if all of those things occur before you can move on to the next step. Okay, another example, cells can't enter mitosis until all the chromosomes have been copied, or cells can't enter anaphase until all the chromosomes have been attached to the spindle fibers. Okay, so not only do we have internal regulators, but we have regulators that come from outside, and this is what we call external regulators. And these are just responses by the proteins from outside events, such as an injury to the cell. And these are signals that tell the cell to speed up or slow down the cell cycle. And an example would be certain growth factors. Okay, so as you're growing up from an embryo in your mother's womb, your embryo is developing. There's growth factors that stimulate the cells to divide, and then also even when you get injured, okay, there's growth factors that stimulate or give signal to the cell and the proteins. Hey, you guys need to start dividing. Okay, so more on external regulators. The molecules on the surface of neighboring cells act as signal to slow down or stop the cell cycle. Remember, I talked about contact inhibition. As soon as the cells come in contact with each other and there's no more room to grow, they're going to completely stop. Okay, so let's go ahead and take a look at some cancer cells. These are some Chinese hamster ovary cells in a culture, in a cell petri dish. And here are some cancer cells. So we're going to go ahead and talk about cancer cells and how that affects the cell cycle. So one thing to note about cancer cells, obviously cancer is very difficult to understand. It's not something that we can completely control, that we completely have control of, but we have noticed that all cancer cells have something in common, and that is the fact that they have lost control of all of their cell division genes. So cancer cells lack contact inhibition. So once they come into contact with each other, they don't think, oh, we need to stop. They have completely overruled, don't have that contact inhibition, and they will just keep dividing and dividing and dividing. And this is, and then all of a sudden they make a huge mass, and this mass is what we call a tumor. Okay, so the cells will just keep dividing and dividing and dividing because somewhere along the cell cycle they have completely lost control of it, and now they don't have any sort of contact inhibition. So they'll keep dividing, doesn't matter how much room, they will just keep packing it and packing it and packing it. Okay, and the tumor is a mass of abnormal cells in normal tissue. Okay, let's look at some characteristics of cancer cells. Like I mentioned, what happens with these cancer cells is that they just don't stop dividing. They just have no control. And it's almost kind of like driving a car that has no brakes. Okay, so what's one of the most dangerous things about cancer cells? Obviously, there are some spots where there's only tumors, and doctors are able to remove those tumors, and patients are able to go into full remission. But one of the most dangerous parts about cancer is that it can actually break off from each other because there's no room. It will go and it'll find room to continue to grow and grow and grow. And so it will start spreading to new places of the body. It'll go into that bloodstream, flow around the body, find a place for it to start dividing, and create more tumors all over the body. And this is what we call metastasis. Okay, so the cells, what happens is one part of the body will get a tumor, and the cells will be spread all over the body, and they will metastasize, or what we call metastasis. Okay, and so there are substances you hear all over the news that cause cancer. All you hear, this causes cancer, that causes cancer. A lot of things cause cancer, and those things that cause cancer are what we call carcinogens. And carcinogens are just things that damage your DNA. So cancer cells are cells that have damage to their DNA, and they have no control of their cell cycle. And that's why they keep dividing and dividing and dividing, because their DNA has actually been damaged. And there are some examples of carcinogens. There's cigarette smoke or tobacco, high levels of radiation. You've probably heard about that when you go to the doctor, when you go to the dentist. Some people only want to get a few x-rays a year. So it's less exposure to radiation, because radiation will damage DNA and could possibly actually cause cancer. There are also a lot of chemicals in the environment. You hear that a lot, mainly from human pollution that eventually cause cancer, and even viruses. Even viruses can cause cancer. You've definitely heard of the HPV virus, which is a cervical virus that is a sexually transmitted disease. And that actually is a virus that can cause cancer, and it's a really, really painful cancer. But we do have vaccines for that. We have what we call the Gardasil shot, and those Gardasil shots actually do prevent that virus from attacking certain areas and causing cancer. Okay, so like I said, cancer is actually quite complicated and can have many different causes. Many things can actually cause cancer, but we know that there's at least one thing in common with all cancer cells, is that they have completely lost control of their cell cycle. So any cancer cells that have damage to a specific gene that we call the p53 gene, and that p53 gene is a gene that actually regulates the cell cycle, the growth of the cell. We also call this the TP53, or tumor protein 53, and this gene codes for a protein that actually regulates the cell cycle. And this protein that regulates the cell cycle actually prevents tumors from forming. So in other words, this is a tumor suppressor gene, and suppressor meaning to stop or to arrest. And so if this gene ends up getting damaged, cells won't be able to respond to the normal cell signals that actually control their growth. Okay, and tomorrow we will talk a lot more about cancer cells and cell regulation. Hopefully that wasn't too long for you. Go ahead and take a look at the different characteristics of normal cells versus cancer cells, and you can see a lot of different things are happening to the cells that have damage to their DNA. And then I want you guys to look at these questions, be able to answer these by tomorrow, discuss these with your partners in class, and we will go over all of these tomorrow. So hopefully that wasn't too bad, and I will see you guys tomorrow. Have a good night.